All right, so we're going to start with chapter 12 today, which is we're getting into the nervous system now. Um, so what happens if you touch something hot, a hot stove, or you immediately pull, right? Question, do you really get burned before you pull? No, it's a reflex, right? And it's a very fast reflex. It happens suddenly. So this quick reaction is possible simply because of the nervous system. So if you think about the nervous system, it's like a high-speed communication grid. That's what it is. So let's look at the jobs of the nervous system. Three main functions. One is it's constantly taking information from the periphery, from the environment, from your body. So it's constantly receiving information, taking in information. Then that information is processed in a center. And this center is called the central nervous system. So that's like the control center. And that consists of the brain and the spinal cord. So the brain and the spinal cord together form the central nervous system. All the rest will be the peripheral nervous system. So broadly, you can divide it into two, central, peripheral, okay? So that's going to be the main division. What's happening in the central nervous system? Decisions are taken. So processing, information is processed in the central nervous system. Let me just move this. And what happens after the information is processed? Some commands are given. Where does the commands go? They go to the skeletal muscles. So in case of if you touch a hot object, your hand moves. That is a skeletal muscle response, right? You are pulling your muscles are getting activated. You're pulling your hand away. So commands can be issued to the muscles, but commands can also be issued to the glands. Okay. So we'll talk about that in a minute, about the commands going to the glands. So let's keep moving forward. Okay, here we go. So we said there are two main divisions. There's a central nervous system and there's a peripheral nervous system. In the peripheral nervous system, there are two functional divisions. One is a sensory, because you're constantly receiving information from the outside, from the environment, and also from your body. So that is the afferent, afferent. That means coming in. And this afferent division is called the sensory division. Then there is a efferent division or the motor division. So if you remember, efferent simply means impulses are going out from the central nervous system. So, or it is exiting. So efferent, exiting. E and E. Okay. So afferent, yeah. Oh, did the slide not change? Oh, so you want to go to the previous one? Yeah. Yeah, you're welcome. I'll be posting this also. Yeah. Ah, sure. Yeah, no problem. Yeah, but I'll post it. Definitely, I'll post it on the. Yeah. Okay. So sensory. So we're talking about the sensory, which is the afferent coming in. Motor, efferent exiting the CNS. Let's go to the sensory. Now, the sensory division has got sensory somatic and also visceral sensory. What do you mean by somatic sensory? Somatic sensory are the signals which are picked from the skin, from the muscles. So if you get hurt, it's going to be your somatic sensory division which receives that information. What is a visceral sensory? Visceral sensory means the impulses from the visceral organ. What is the viscera in the body? Internal organs, what are they? The urinary bladder, when you feel the bladder is full, you will feel, you know it, right? You have to run to the restroom. Or if you're hungry, you'll feel it. You're thirsty. These are the visceral sensations. Again, if you look at the motor also has got the same thing, visceral motor and somatic motor. So somatic motor, they control the voluntary muscles. What are the voluntary muscles? The skeletal muscles, which are under your control. They are the voluntary muscles. So the somatic motor controls the voluntary muscles, and the visceral motor controls the involuntary muscles. What are the involuntary muscles? Your heart muscles, the muscles in your stomach, intestine, all those are involuntary muscles. And they come under the category, come under the visceral motor come under a category called autonomic nervous system, which will be one of the topics we are going to learn. So autonomic nervous system is what we call the visceral motor division. Okay? And that has got two divisions. There is a sympathetic division and there is a parasympathetic division. Sympathetic, in other words, is called the fight or flight. That's when you're in a dangerous situation. It prepares the body for action. What happens when you're in a situation like that, where you're in trouble? Your heart rate goes up, your respiratory rate goes up. Okay, your body is getting ready for fight or flight. So that is the sympathetic division. And then there is a parasympathetic division. The parasympathetic division is when you're relaxed, rest and digest. So when you're relaxed, you are in the parasympathetic, under the parasympathetic division's control. So your heart rate goes down, you're totally relaxed, your digestive system is now active. A lot of digestive juices are produced. The glands produce a lot of juice. Your digestion is in full swing. Your urinary system is also now relaxed. But if you're tense, sometimes you may not want to go to the restroom, right? Because the parasympathetic is not working there at all. That's when you're relaxed. So these are the two divisions of the autonomic nervous system, sympathetic and parasympathetic. And you know the functions of each now, right? Okay. Now, neurons. Neuron is the main players in the nervous system. The neuron is the basic unit of the nervous system. There are three types of neurons. You have a sensory neuron, which we discussed. They bring in signals, right? So are they afferent or efferent? Afferent, yes. Afferent, okay. And then there's the motor neurons, which are sending out the signals. They are efferent. And in between, there is something called interneurons. These are the neurons which link the sensory and the motor neurons. These neurons are inside the central nervous system, which is the brain and the spinal cord. They are the main decision makers. That's where the decisions are made. Others carry the information. Come on. Okay. So for doing all these things, the neurons have certain properties. One is it's excitable. A neuron has got the ability to respond to a stimuli. Number two, if when a stimulus is there is a change, something happens, and that change is called excitability. And not only that is excitable, that is conducted. It sends the electrical signal along their length. So it just doesn't stay there. So if your hits are here, then you suddenly you may pull back initially, you may feel the pain. All these things happen because you're receiving information from the periphery to the central nervous system. So it gets excited, it sends the signal to the central nervous system, and then something else happens. There's secretions happening. It releases chemicals, and these chemicals are called neurotransmitters. And we are going to talk a lot about neurotransmitters because that's exactly what causes all the changes. So the nerve cells get stimulated, they pass that impulse. How do they pass it? Through neurotransmitters. So the neuron sends so initially the signal, when a neuron is stimulated, it's an electrical signal. But when it passes it on to the next neuron, it is a chemical signal. So we're going to go talk a lot about these signals and how are the signals being transmitted, what's happening there, what is memory, what is learning, all these things. Okay? We don't, so I'll try to summarize everything so that you get a gist of the whole thing. But as I told you, I won't have the time to go through everything, but that's your job. You have to go through the readings, okay? But when I finish my class, it'll give you a stimulation, it'll give you a basic idea about what it is, okay? So that's my job here. So let's have a look at the structure of a neuron. So the neuron consists of a cell body. This is a cell body, and the cell body is called soma, S-O-M-A. So this is a cell body. And from the cell body, there are some processes. You can see some processes, right? So there are two types of processes. These short processes here, they are called the dendrites. And there's one long process here, that's called the axon. And in the cell body, there is a nucleus. And inside the nucleus, there is a nucleolus. And there are some dark areas here. They are called the Nissl bodies. They're actually the endoplasmic reticulum, or the rough endoplasmic reticulum. Just like any other cell. It contains all the organelles. But the difference is these two processes, the dendrites and the axons. So what are the dendrites? Dendrites are the shorter process. Axon is large. Dendrites receive the impulses, whereas axon, they're longer than the dendrites. The axon send out the impulses. And the area where the axon leaves the cell body, this area is slightly thickened. And this area is called axon hillock. So there's an axon here, and this little widened area at the beginning is the axon hillock. It's like a trigger zone. So that is, if an impulse is of a certain magnitude, it will fire and it will go all the way down the axon. So that's where that trigger zone is located. That is called the axon hillock. So if you imagine a nerve of the leg, that may have a very long axon, maybe a meter long. So there's a lot of length there. So, and the cells need nutrition, and the cells are producing a lot of proteins inside, and all of this has to move up and down, right? All the proteins, nutrients, everything has to go along the axon. And this is what we call the axonal transport. So there's two types of transport. One is moving stuff from the cell body into the axon. That is called the anterograde transport. And there is a retrograde transport, which is bringing all the waste back to the cell body. So it's not like, okay, there's a big extension there and well, it'll take care of, no, the cell body is minding it. So it's under the control of the cell body. And there's a very active two-way transport system happening in the axon. Let's look at the structural type of the neurons. I told you about the classic neuron. This is called the multipolar neuron. So that's what we were discussing so far when we studied the structure. So the multipolar neuron is the most common type of neuron. And when you think about a neuron, that's what comes to your mind, a multipolar neuron. But there are other types of neuron. The multipolar neuron, the features are, it has got one axon and it has got multiple dendrites. Where is it seen? In the brain and the spinal cord. Why do you need a lot of dendrites and only one axon? Because it's receiving impulses from various sources. It's coming to the cell body, and from the cell body, it's sending impulses to the other cells or the next neurons. There's a lot more happening than what we think, and we'll come to that. Let's look at another type of neuron, a bipolar neuron. So this is a bipolar neuron, which means there is one axon and one dendrite. So here you can see one axon, one dendrite. And this is particularly seen in the eye, in the special senses. That's one of the chapters. That's the last chapter, special senses. So you'll be learning about the special senses, especially the eye, the ear, the olfactory cells. What do you mean by olfaction? Smell, yes. So that's the olfactory cells. And then you have unipolar neurons. These are the unipolar. That means there's a single process which leaves the cell body. It splits like a T. One goes to the periphery, one goes to the central nervous system. That's a unipolar. And these are particularly designed for fast transmission of impulses. For example, pain, touch, pressure, all these things. So it has a fast transmission rate. And there are some neurons with no axons. So here you can see a neuron. Oh, okay. This neuron here does not have any axons. It only has dendrites. And this is a type of neurons, this is called an anaxonic neuron. And these are seen in the retina of the eye. Signals are not sent out like the multipolar neurons. They are processed there itself. So that's another type of neuron. We're not going into a lot of details, but I want you to understand there are different types of neurons. And function follows the form. So there's a reason for it, why these neurons are made different. Okay. So in the nervous system, we were talking about the neurons so far. Is there anything else in the nervous system? In the lab, we discussed it. The glial cells, right? So the nervous system has got neurons and glial cells, or it's called the neuroglia, okay? Neuroglia are really the supportive cells, and they are much more numerous than the neurons, like 50 to 1. So for every single neuron, there are 50 glial cells. How many neurons are there in the body? Approximately 1 trillion. Yeah. So the neuroglial cells are 50 times more. Okay. That is 50 trillion. So there are 4 types of glial cells or neuroglial cells in the CNS. One main one is the oligodendrocytes. Here you can see an oligodendrocyte. And what it does is it makes myelin. Some of the nerves are coated with a covering, like an insulation. And this insulation is called myelin. It's a myelin insulation. And a nerve which has got a coating of myelin is called a myelinated nerve. And this myelin is produced by the oligodendrocytes. Why do nerves have myelin? To increase the conduction speed. And then there are some other glial cells called ependymal cells. These are the cells which secrete a fluid around the brain. And this fluid is called the cerebrospinal fluid. It protects the brain. That's why if you get a hit, there's a cushioning happening because of this fluid. It's called the cerebrospinal fluid, CSF. So if somebody complains of, say, headache, difficulty, high fever, difficulty seeing, all these things, a doctor can suspect meningitis, which is inflammation of the brain. And if you have meningitis, the diagnostic test is to tap the CSF and to send it for examination. So CSF is produced by the ependymal cells. And then there are some defense cells. They are the microglia. These are the cells that fight infection. Yeah.
Is there cerebrospinal fluid in the spine?
Yes. Cerebrospinal fluid is all around the brain, and the spinal cord is continuation of the brain, right? So through the foramen magnum, the brainstem comes there. It continues as the spinal cord. And the spinal cord is also covered with the meninges, and it has got the cerebral spinal fluid. And also in the center of the spinal cord, there is a little hole, and there is fluid, sort of the CSF or the cerebrospinal fluid passing through it. So it's all over the central nervous system, brain and spinal cord. Astrocytes, that's another important cell. It's a very busy cell. It's here, you can see this is an astrocyte. Kind of a star-shaped cell. Actually, if you look under the microscope, which you probably have seen in the lab, it looks like a spider. If you see it once, you won't forget it. Okay? So that's what an astrocyte looks like. And that's a favorite question in the lab exam. So you may want to have a good look at it. Astrocyte has got some other major functions. It produces what's known as a blood-brain barrier. So you can see these are the blood vessels in the brain. And these blood vessels are actually covered by the astrocytes. They put the foot on a cell and they bring it together. They create a blood-brain barrier. And this blood-brain barrier is good because it prevents any bacteria creeping into the brain. So there's a lot of capillaries, and capillaries have got gaps in their endothelium. Endothelium are not thickly packed, but the astrocytes make them close and prevents any ingress or any exit of the microorganisms. So it creates a blood-brain barrier. Yeah.
So the astrocytes squeeze all the nutrients out of the blood?
Not nutrients, the cells together and prevents the gaps. So it's got nothing to, it's got another function with the nutrients. We are coming to that. But for now, I want you to understand it creates a blood-brain barrier. So this blood-brain barrier is good because it prevents any microorganisms coming into the brain. But there's also another disadvantage. If a person has got a tumor, a cancer of the brain, you may want to give some chemotherapy, right? And chemotherapy does not enter the brain. So if you have a malignant brain tumor, chemotherapy is out of question. So we just discussed this in the last lab. So if you have a malignant brain tumor, chemotherapy cannot be used, but what are the other modalities of treating cancer? Radiation? Measles? For brain tumor?
Into like little black donuts and throw
Really? I've never heard of that. Yeah, measles is a virus.
them.
Yeah, so I don't know, probably that's something new. But radiation is one of them, and the other one is surgery. Remove that part, which is, you know, if you remove a part of the brain, that's quite damaging. So radiation and surgery are the mainstays. But the astrocytes have got some other functions. For example, we were talking about nutrients. Yes, it provides nutrients, and also it also changes or regulates the chemical environment. So it is a very busy cell. Now, if you think of it, we were just talking about brain tumors. Brain tumors, can they come from neurons? No, they never come from neurons. Why? Because neurons don't divide. They don't multiply. And for cancer, you need a cell that multiplies fast. Uncontrolled multiplication is what cancer is. So here, the mature neurons don't divide. So brain tumors or cancers of the brain can never come from a neuron. So where does it come from? Sean said, yeah, that's a glial cells. From the glial cells. Glial cells can divide. So the glial cells, when you said Schwann cell, you're talking about one type of glial cells. But I'm talking about all the types of glial cells. We'll talk about Schwann cells. But for the time being, I want you to know that all the glial cells are capable of division and they can produce tumors. And glial tumors is one of the most common tumors in the brain. Glial tumors are fast growing and they are malignant. But chemotherapy is difficult because of the astrocytes. The astrocytes produce a blood-brain barrier. Okay, now coming back to what is this little structure here? That's an oligodendrocyte, okay? So we talked about the oligodendrocyte. We said the oligodendrocyte produces myelin, correct. So the myelin sheath is like an insulation. So if you have a wire, you have a plastic coating outside the wire. We call that the insulation. And the myelin sheath is an insulation around the axon. Why do you need a myelin sheath? To speed up the conduction velocity. That's the main purpose. So now, what produces the myelin in the CNS? What produces it in the PNS? CNS is central nervous system. So central nervous system, it's produced by the oligodendrocytes. But in the peripheral nervous system means exactly anything outside the brain and spinal cord. That in the peripheral nervous system, Schwann cells produce the myelin sheath. Okay, so that's a big difference between, and this is a very, very important thing because we are going to come into some other major stuff very soon. So what is this myelin sheath? It's like a fatty thing. And it contains about 80% of lipids, which means it's basically fat, and 20% protein. And that process by which myelin is formed is called myelination. And we said myelin is mainly fat. That means fat is necessary for production of myelin. When is myelin produced? It starts at a very young age. And it's slowly building up. It's producing all this coatings over the neurons. This process starts at around 14 weeks of intrauterine life, IUL, and it continues up to about 20 years till adulthood. So this is why we will never restrict fat for kids. For our children, infants, they need a lot of fat because they are in that stage of myelination. That's why dietary fat is very important for babies and children. Now, if you look at a myelin sheath, you see that this sheath is segmented. You see there's one segment here, the next segment, maybe here, there's a picture here. You can see there's one segment here, and then there's a gap here, another segment, a gap. That's how it is. And these segments, or these gaps between the segments, are called nodes of Ranvier. There's probably a guy, the guy's name was Ranvier, I don't know, but it's called the nodes of Ranvier. Another favorite question in the practical, nodes of Ranvier. And we said, in the peripheral nervous system, myelin is produced by Schwann cells. So what does a Schwann cell do? It comes around the axon and it starts wrapping around the axon. And then once it wraps, the outermost layer is where you see the nucleus. This layer, the outermost layer of this wrapping, is called neurolemma. Very important. Neurolemma is crucial for nerve regeneration. Now, you might have heard about the accident which happened to Superman. He fell off, it was a while ago, a horse. And there was a damage to his spinal cord, right? A cervical area. And he became totally paralyzed. So he was on a kind of a wheelchair, which was, and he had to keep his neck up because there was a big fracture here and the whole area was paralyzed. He did not recover. Why? Because in the central nervous system, regeneration does not happen. Why? Because in the central nervous system, who produces the myelin? Oligodendrocytes, correct. They produce myelin, but they don't produce neurolima. And neurolima is absolutely essential for nerve regeneration.
Neurolima has the nucleus to what?
Nucleus, I mean, that is where the nucleus resides.
Oh, the next cell is gonna be?
Oh, no, this is the nucleus of the cell, the Schwann cell. The Schwann cell is kind of wrapping around, and the outermost layer is where you can see the nucleus, and that outermost layer is called the neurolima, which is not seen in a oligodendrocyte. That's why regeneration does not happen in the brain and spinal cord. But it is possible in the peripheral nerves. There's one more thing which I have to talk. It'll come in a second. So I want to talk about a couple of diseases. One is multiple sclerosis, MS. What is multiple sclerosis? It's an autoimmune disease. When you say autoimmune, that means the body is recognizing something as foreign and attacking it. Here, the myelin sheath is considered foreign. The body thinks the myelin sheath is foreign. It attacks it, and it attacks the oligodendrocytes. The insulation is damaged. The patient ends up with numbness, loss of balance, muscle weakness, visual changes, and it's very debilitating. So that's multiple sclerosis. And there's another disease called Tay-Sachs disease, which is a hereditary disease. It is not, it's something different. The mechanism is different. Here, there's a buildup of abnormal fatty acids because an enzyme is missing. And this disrupts the nerve function, and the patients usually have got visual problems, loss of coordination, blindness, and usually it's a very fatal disease. So these are the two diseases which, I mean, I just want you to know about it. So, okay, coming back to our nerve conduction. Now, I told you nerve conducts impulses, right? That's one of the main functions of the nerve. It passes the impulses through the axons. So, what are the two factors that controls the speed of impulses? Number one is the diameter of the axon. So if you have a larger axon, the signals travel faster. The second factor is presence or absence of myelin. If you have myelin, it's faster. If you don't have myelin, it's slower. So the speed difference here is if you have a small neuron, I said if you have a thick neuron, it's faster, right? If a small neuron, no myelin, it's approximately 0.5 to 2 meters per second. That's the signal speed. Whereas if you have a small neuron with myelination, then suddenly it goes up to 15 meters per second. But if you have a large neuron and if you have myelin, it suddenly jumps to 120 meters per second. That's highway speeds. So why do you need all these fast transmission? Because you have to have quick reactions. You may have a situation where you're touching something hot and the brain has taken, even before you know it, you pull your hand away. Because actually in that case, the impulse don't reach the brain. The spinal cord has already taken a decision, executed action. And later the brain will come to know about it. That's called a reflex action. And we'll study that later when we reach the spinal cord. But for the time being, let's keep ourselves to the central nervous system and the peripheral nervous system. This is the slide which I wanted to show you. So if a nerve is damaged, now we're talking about the peripheral nerve, okay? Say I have, say the radial nerve or any of the nerves in your hand. If it is damaged, can it regenerate?
Yeah.
Yes. Why? Because it has got neurolemma. Schwann cells, they wrap around. The outermost layer is called the neurolemma. So but there's one more condition. The cell body has to be intact and some neurolemma must be present. If these two conditions are there, there's a high chance that, see here, you have a trauma, it's cut, but the neurolemma is, let me just move it. Yeah. So the neurolemma here kind of produces a regeneration tube. Okay. So this regeneration tube will guide the growth of the axon. So that's what's happening. Whereas in the central nervous system, it is not possible because there's no neurolemma, no regeneration tube is produced, and also there are chemicals released by the glial cells that inhibit the axonal growth. So there are many reasons why it doesn't happen in the central nervous system.
Nerves regenerate with schwannomatosis.
With what?
Schwannomatosis.
Say that again?
Schwannomatosis. It's a nerve disorder that causes tumors.
Okay, no, I don't know. No. Yeah, but the first part of your question is, can nerves regenerate? Yes, in the periphery, provided the cell body is intact and there is some neurolemma available. In that case, it can produce a regeneration tube and axonal regeneration. So did you say schwannomatosis? Matosis, right. Yeah, I have to look into that. Okay, now we were talking about impulses. What are these impulses? The impulses are actually electrical impulses. So a neuron, even at rest, has got an electrical impulse. That means there's something called a resting membrane potential. So that means there is a difference in the charge. Usually the outside of the cell is positive, inside of the cell is negative. So if you put a microvoltmeter on a nerve cell, you can see that there is a voltage there. The outside is always positive, inside is negative. Why? Because there are pumps. And there's one major pump. We talked about this when we were studying muscles. Now we'll go into some more details here. We talked about the sodium-potassium pump. Now, this pump pushes sodium out, and there are potassium pumps. It pushes potassium in. But there's a difference. It pushes 3 sodium ions out, and at the same time, it pushes only 2 potassium ions in. So 3 on the outside, 2 on the inside. So the outside is more positive. Inside is less positive. That is why when you put a microvoltmeter, it appears like the outside is positive and the inside is negative. It's a differential. And the voltage which is recorded is minus 70 millivolt. But these pumps just don't work for free. They need energy. And what is the energy for the cells? ATP. So it is taking energy. So even at rest, it is consuming ATP. Now I want to talk about a local potential. So a local potential, say you stimulate a sensory nerve, okay? There will be some changes happening in the cell membrane, and it is basically changes in the sodium-potassium pump. Usually the sodium-potassium pump will stop, and the sodium channels, which are normally closed, opens. Sodium, which is heavily concentrated on the outside, starts flowing into the inside of the cell. So here you can see sodium has started entering. What is the charge of the sodium? Positive. So the inside is minus negative, minus 70 millivolt. Sodium ions, positively charged ions, are entering. What happens to the charge from minus 70? Where does it go? Yeah, it goes up, which means it goes to minus 60, minus 50. It goes up, right? Yeah, because it's a positive charge. So in other words, it's becoming less negative. So here you can see from here, it is reaching here. And at this point, at minus 55, that is a critical level. That is called the threshold level. So at that threshold level, it will fire. And once it hits here, lots of sodium channels open. Suddenly, it starts huge number of sodium channels open, which means it will pull it up like this. It will go from minus 70 to minus 55. Sometimes it will reach 0. It will go all the way up to plus 35. Once it hits plus 35, then what happens is that the sodium channels close. So there is a movement from minus 70 to plus 35. This is called depolarization. And then once it reaches here, the potassium channels will start opening. And then it will start going down, and this is called repolarization. Let me move to the next slide. So here it already reached plus 35, and I told you the sodium channels close. They were open all this time. Potassium channels now open. And what does potassium do? It is going into it starts exiting. Normally it's on the inside, right? But here it starts exiting. So when it exits, what is it doing? Potassium is positively charged. So it brings all the positive charge is going out. So from plus 35, it starts going down into the negative territory. So minus plus 35, plus 25, 0, minus 10, 20, 30, 40, it reaches minus 70. But it actually overshoots a little bit. So let's stop before the, the overshooting. Say it reaches minus 70, and this part here is called the depolarization from minus 70 to plus 35, and from plus 35 to wherever it is reached. I told you it overshoots a little bit. That is called repolarization. So for a brief period, it overshoots. It becomes a little more negative. Maybe it'll go to minus 75. And this is called hyperpolarization. But later, all these channels will start working. The sodium pump, the potassium pump will start pushing ions in and out, and it restores the balance. It'll come back to minus 70. So this is what we call an action potential. So we were talking about local potential and action potential. So there's a little difference there. So a local potential is when there is a stimulus and slowly the charge, which is minus 70, it starts going down. It starts moving towards an action potential, but it does not really reach the threshold. So when it doesn't reach the threshold, we call that a local potential. If it reaches the threshold, we call it a action potential. So if you imagine a local potential like a ripple in the water, action potential is like a massive wave. It goes all the way up to here. But a local potential will kind of stop before the threshold. Clear? So that's the difference between a local potential and an action potential. There are some other major differences. One is, I told you, in an action potential, threshold is reached. In the local potential, it is not reached. And in the action potential, once it reaches the threshold, it fires. Then you can't stop it. It's called an all or none law. If it happens, it goes to the full blast. Local potential also is by ligand channels. Ligand channels are chemical differences. They are chemical channels. Whereas the action potential is voltage gated channels. They are the sodium and potassium channels. So there are different types of channels. But what I want you to understand is action potential follows an all or none law. The other one does not develop into an action potential. It is a sub-threshold. It doesn't reach the threshold. Refractory period. Refractory period means that is a kind of a reset time for the nerve. So during this period, another action potential is not possible. So there are two types of refractory period. There's an absolute refractory period, which means it is impossible to fire another impulse during that time. So this is the period when the sodium channels are opening. So it starts opening here, and it's over here, it slowly opens. Once it hits the threshold, all the channels open, a huge number of sodium channels open. depolarization happens. So during the opening of the sodium channel, there's no way you can have another action potential. And then the sodium channel is still open up to here. When it reaches here, it starts. That's when the repolarization is almost finished. Then the sodium channels close, and then the potassium channels open. So up to here, from here to here, we call it the absolute refractory period. And after that, you have a relative refractory period. Here, the sodium channels are closed, but the potassium channels are open. And at this stage, if you produce a higher stimulus, a stronger stimulus, you can activate an action potential. And why do you need this refractory period? Is it of any benefit? Yes, there is a benefit. The benefit is, one, if this is a nerve, okay, there is an action potential happening here. That action potential is going to stimulate another action potential, which stimulates another one, and that's how the nerve impulses are moving. When there's a refractory period, that means this is activated, this has reached here, but this part is now in the refractory period. So impulses cannot go back. So the impulses can only move forward. So refractory period is the reason why nerve impulses move in one direction. So now you know how a nerve impulse moves, right? There's one area here which is now depolarized. Okay, the next area becomes depolarized. It's like a set of dominoes. It starts here, then it all starts falling one after the other. So that's how an action potential moves along a nerve. So here you have one area, second area is this is now depolarized. Next area is depolarized. It keeps moving forward. It doesn't go back because of the refractory period. So now we're talking about what kind of a nerve here. This is an unmyelinated nerve. Okay, there's no myelin there. In a myelinated nerve, it's a different scenario. Let's look at that. In a myelinated nerve, I said that there are gaps in the myelination. These gaps are called nodes of Ranvier. So these nodes of Ranvier are where there are a lot of there's a lot of the channels. They are concentrated here. Voltage gated channels, that was the word I was looking for. So the voltage gated channels are actually concentrated at the nodes of Ranvier. So what happens is that the signal jumps from one node to the next node. It's not like the unmyelinated nerve. In the unmyelinated nerve, every single segment has to be depolarized, repolarized, depolarized, repolarized. So every segment has to go through the process. But in a myelinated nerve, it jumps. That's why it's called saltatory conduction. Saltare is it Latin? It means jumping. So it's jumping from one node to the other. That's what you saw in that particular, see here, it's jumping from here to here. See how fast it is. Because it's jumping from node to node. Whereas over here, it's moving along the length of the nerve. So in myelinated nerve, the conduction is called saltatory conduction. And we talked about the speeds. Okay. So far we were talking about the action potential and the impulses in one nerve or one axon. So one axon, what does it do? It talks to another axon, right? Impulses has to move all the length of the nerve. And we only talked about one neuron or one axon. Okay, so what is a synapse? The meeting point where one neuron talks to another neuron is called a synapse. So it doesn't have to be a neuron, it could be a muscle. We learned about a nerve touching a muscle, right? What did we call that junction? Nerve, muscle, neuromuscular junction. That was in the muscles. When you're in the muscle lab, you saw a model where a neuron is coming, is attaching to a muscle, and we call that the neuromuscular junction, right? Okay, so that is another connection. That's a meeting point. But in case of a nerve, the first neuron in the path is called a presynaptic neuron. The connection is called a synapsis. So there's a presynaptic neuron, postsynaptic neuron. And we said the signal moves only in one way, does not go back. Always from the presynaptic to the postsynaptic. And it can happen in different places. It can be between an axon and a dendrite. It can, so here, this is an axon. It's connected to a dendrite. It could be from an axon to a soma, to the cell body. Or it can be from the axon to another axon. So it can be axodendritic synapse, axosomatic synapse, axonic synapse. Any synapse is possible. So what happens at the synapse? We studied this when we studied muscle. What's happening here? So you have a, what is this called? This is the axon, and what is this bulge called? Axon terminal. What does it contain? Acetylcholine. Where is the acetylcholine? Is it, it's lying loose, or is it vesicles? So there are synaptic vesicles, and that contains, they are like bubbles. They contain the neurotransmitter acetylcholine, and these are called the synaptic vesicles. And what is this? Across the synaptic cleft, there is the second neuron. We call that the postsynaptic neuron. Okay, and this is where we said a chemical synapse happens. So initially it was an electrical synapse. Now it is a chemical transmission. How does that happen? Because acetylcholine, which is a chemical neurotransmitter, is released into the synaptic cleft. It moves through the synaptic cleft and it attaches to the membrane here. This membrane has got receptors and they bind to the receptors. And that's why it's called a chemical synapse. So electrical, initially it's electrical, then it becomes chemical. And this is what is really causing us to, our memory, all these things are based on this chemical synapse. In mammals, they have very little chemical synapse. Humans have a lot of chemical synapse. That's why we have higher thinking. Can you believe it's just that chemical synapse which is giving all the complexity, all that thought process, all those higher functions is because of the chemical synapse. So that's serious stuff, right? So let's have a look into it. So what's happening? Let's have a look at the at a very, very basic level. So an impulse comes here, and we said there are synaptic vesicles. It contains neurotransmitter. So when an impulse reaches here, calcium ions are released, and the calcium ions will bind to these vesicles, and the vesicle will kind of have an exocytosis. So the membrane of the vesicle attaches to the membrane of the cell membrane here, and the content is pushed out. And when this content, what is the content? It's a neurotransmitter, acetylcholine, ACH, acetylcholine. So it's pushed out, and the acetylcholine will come and it will attach to the receptors here in the postsynaptic neuron. The main neurotransmitter is acetylcholine. There are other neurotransmitters, and we'll be talking about that. Oh, here it is. So some of the neurotransmitters will be stimulatory. It stimulates, or it is excitatory. Some neurotransmitters are inhibitory. What are the excitatory neurotransmitters? Acetylcholine is one of them. Epinephrine, norepinephrine. There are hundreds of neurotransmitters. What are the inhibitory? I want you to know a few, just GABA and, yeah, I think I've only put one, GABA. What is GABA? GABA aminobutyric acid. So I told you, these are considered to be excitatory, but sometimes the same neurotransmitter can be excitatory in one particular situation, and it could be inhibitory in another situation. For example, acetylcholine, we said, is an excitatory neurotransmitter, which is correct. But if it is attached to a cardiac muscle, it is inhibitory. So acetylcholine excites a skeletal muscle, but acetylcholine will inhibit the cardiac muscle. So if you have different types of, it all depends on the type of receptors. Now, a synapse which is using acetylcholine is called a cholinergic synapse. A synapse which is using epinephrine and norepinephrine, they are called the adrenergic synapse. This is a whole big chapter, but I want you to at least know this, okay? So if you have acetylcholine as the neurotransmitter, we call that a cholinergic synapse. Epinephrine, norepinephrine is called a adrenergic synapse. So far we were talking about neurotransmitters. They go and stimulate the postsynaptic cell. But sometimes it's not a direct action. Sometimes it's an indirect action. Oh my God, it's already 5:40. Okay, I have to move a little fast. So here we talked about all this, and we talked about some of them are excitatory, and we said some of them are inhibitory. And why is it excitatory and inhibitory? It's simply because we said there's a depolarization happening. From -70, it moves up to -55. At -55, it fires. So that's excitatory. But if you move in the opposite direction, say you have an inhibitory signal, and these signals, if they cause the voltage to go from -70 to -90, that is an inhibitory. And that is what chloride channels are doing. So if you excite a chloride channel, the chloride channel is negative, it pushes it down, it moves away from the threshold. So this will be a inhibitory. So acetylcholine is considered to be a excitatory neurotransmitter, and it produces a EPSP, excitatory postsynaptic potential. That means a threshold can possibly reach. Another one is epinephrine, norepinephrine. They also produce excitatory impulses. Whereas another one is epinephrine, norepinephrine, also produce excitatory, but they have a difference. They don't stimulate directly. What they do is that they stimulate or they activate what is known as a second messenger system, such as cyclic AMP. So this is called this is another link which is added to the equation, which means now it's going to be a little slower, right? Because you have to one more link in the loop. So it may be slower, but there's something called enzyme amplification, which means a single molecule of epinephrine or norepinephrine can stimulate a large number of molecules. So this is what happens in a fight or flight situation. There's signal amplification. In the fight or flight, epinephrine, norepinephrine, they are the neurotransmitters.
Adrenaline.
Adrenaline, yeah. Adrenaline is what we call epinephrine. That is like that they are the same. Now, GABA is an inhibitory neurotransmitter. What it does is it will move it away from the threshold. So the postsynaptic neuron is less likely to fire an action potential. So this is what we call a IPSP, inhibitory postsynaptic potential. So how does a signal end? A signal ends when the neurotransmitter stops functioning there. So there should be either the cell should stop producing the neurotransmitter, or the neurotransmitter which is already there must be cleared in the synaptic cleft. And what are the mechanisms? There are a few mechanisms. One is whatever is there can simply diffuse out. So it's all gone. Now the impulse has stopped. Or there could be a degradation of the neurotransmitter. And usually there is an enzyme called acetylcholinesterase, which breaks down acetylcholine into acetate and choline. And this choline is actually recycled. So that is a degradation process. Or there can be a reuptake, which means the neurotransmitters are pumped back into the axon terminal. So if there's neurotransmitter present in the synaptic cleft, the firing will keep happening. So we have to move it away. Either it diffuses away, or it's degraded, or it is pumped back into the axon terminal. Okay, so there's a few more slides. I'll post that on as part 2. Because I think, what time do we finish normally? We have 5 minutes? Oh, then I can finish it. Yeah, there's only 5 minutes left. Okay, so so we were talking about neural integration. That means the brain or the nerves are receiving impulses from multiple sources, and they are taking all these impulses and they are producing action potentials and they are firing. This is what we call neural integration. So once it adds up all the incoming impulses, it decides whether to fire or not. And they are chemical synapses. Chemical synapses is the key. That's where the decision happens. There's nobody sitting there and taking a decision. It is simply chemicals, which is how these decisions are taken. So you can have impulses coming from multiple sources. They converge onto a neuron, and then all these neurons will produce impulses. They converge onto another neuron. For example, for respiratory system, when you're breathing, you have impulses from various parts of the body. You have impulses coming from the carbon dioxide center, you have from the oxygen center. All these impulses come to the brain, and then you're producing a rhythm of breathing. That's a converging circuit. A diverging circuit is the opposite. One neuron talks to many neurons. That's what happens when you're walking. So the brain will produce an impulse that goes to multiple neurons. That goes to multiple neurons. Many muscles are active when you're walking or running or whatever. Then there are reverberating circuits, which means there are repetitive signals like a loop. So that's when breathing is a typical example. You're not thinking about breathing, right? You just breathe. It's like a loop. So in coding language, they call it a loop. If you want something to keep going like again and again, so that's what happens. Or it can be a parallel after discharge, which means initially there is a divergence and then there is a convergence. For example, and there's a delay happening between all these synapses. For example, if you take a picture with a flashlight, sometimes that flash you'll keep seeing that for a few seconds, right? That is a typical parallel after discharge circuit. Because due to the synaptic delay, the postsynaptic neurons produces a output even after the input has stopped. Memory and learning, that's the last slide, okay? So bear with me for 2 minutes. So memory is not a place where things are stored. What is memory? Memory is actually a pathway. And synaptic potential means you're strengthening the connection. For that, people use different techniques. Some people use, they will revise it often. Some people use flashcards. Some people use mind mapping. You do different techniques, but what you're doing is that you are strengthening the connection. You're making that pathway strong. So that is the key mechanism happening in memory. And a couple of diseases, one is Alzheimer's. Everybody knows that when there is progressive memory loss. Why is it happening? Because there's a buildup of amyloid plaques, neurofibrillary tangles in the brain. They're kind of a situation where the brain shrinks. That's Alzheimer's. Treatment is very challenging. I know a lot of people who have Alzheimer's, and it's really sad. My colleagues' parents, and it's terrible. They don't even recognize them. So they'll say, "Who are you?" to their own son. So Parkinson's is another balance disorder. That is because the dopamine producing neurons die. And the treatment is to replace the dopamine. They give a tablet called levodopa, but that doesn't reach the brain. That's a big problem. So these are the two diseases which are Alzheimer's particularly is now increasing a lot, and it's very worrying now. Okay, so that's it. Thank you. Oh, next week is the exam. Okay, don't forget.
Here, not on that.
Say that again.